You know that pill in your medicine cabinet? Ever stop to think about how it got there? It's not just a simple story. It's this incredible journey of science, biology, you name it. And that's exactly what we're going to unpack today. How we get from a single molecule all the way to a life-saving drug.
All right, to get us started, let's kick things off with a bit of a medical mystery. Picture this. Someone overdoses on aspirin, which, chemically speaking, is an acid. The treatment? Sodium bicarbonate, a base. I know, it sounds like something straight out of high school chemistry class, right? Acid plus base. But this is a real life-saving technique. And stick with me because by the end of this, you're going to know exactly how this clever trick works.
So to crack that aspirin puzzle, we've got to go back to basics. I mean, what even is a drug? How does some random chemical get this incredible power to tweak our biology in such a precise way? Let's dig in.
And that question brings us right to the heart of pharmacology. You can think of it as the science of how drugs and our bodies, well, how they talk to each other. These substances are like little chemical messengers, seeking out super specific targets in our system. And when they find their target and bind to it, they act like a light switch, flipping a process on or shutting it right down.
Okay, so for any drug to actually do its job, it has to check three really important boxes. First up is size. It's got to be Goldilocks, small enough to move around the body, but big enough so it doesn't just bind to everything it bumps into. Second, shape. This one is huge. The drug has to fit its target like a key fitting into a very specific lock. If the shape is wrong, nothing happens. And finally, we've got the bonds it makes. You might think stronger is better, but actually, drugs usually form weaker bonds, and that's a good thing. It means they're selective, they hit their target, and they don't get permanently glued to the wrong places.
This all leads back to one of the most fundamental rules in all of medicine, something a physician named Paracelsus figured out like 500 years ago. He said the dose makes the poison, and it's so true. Literally anything can be harmful if you have too much of it, even something as simple as water. It's all about how much. It all comes down to the dose.
Okay, so now we have a pretty good idea of what a drug is. But here's the thing, it's not a one-way street. Our body isn't just a passive bystander here. It fights back. It processes. It interacts. This is where we get into how the body handles these foreign substances.
Yeah, this interaction is totally a two-way street, and scientists have split it into two big ideas. The first is pharmacokinetics. A simple way to remember it is what the body does to the drug. It's all about how your body absorbs the drug, moves it around, and eventually, gets rid of it. The other side of the coin is pharmacodynamics, and that's what the drug does to the body. This is the part we usually care about, the actual effect the drug has. And right here, this is the key to solving that aspirin puzzle we started with. See, for a drug to get pretty much anywhere, it has to cross through cell membranes. And you can just think of those as these little fatty walls. It's kind of like trying to mix oil and water. Charged molecules, they get bounced right off, but uncharged molecules, they can sneak right through. So what's the one thing that can flip a drug's charge from on to off? The pH of whatever liquid it's floating in.
Okay, so here it is, the big reveal. Aspirin is an acid. When someone overdoses, their blood becomes more acidic, which keeps the aspirin molecules uncharged. That means they can slip right through those fatty cell walls and get into tissues where they cause a lot of damage. But here's the genius part. Doctors give sodium bicarbonate to make the urine very alkaline, or basic. And in that basic environment, bam, the aspirin molecule gets a charge. Now it's trapped. It can't cross back out of the urine into the body. It is literally stuck in the pipeline to be flushed out. This cool little trick is called ion trapping.
All right, so we figured out how the drug gets where it needs to go, but what happens when it actually gets there? Now we're talking about pharmacodynamics, the moment that key clicks into the lock.
When a drug finds its target, its receptor, it usually plays one of two roles. You've got your agonists, which are the activators. They're the key that not only fits the lock, but turns it, flipping that biological switch to on. Then you have the antagonists. They're the blockers. Think of it like a key that fits in the lock but is broken off, so it can't turn. It just sits there, blocking the real key from getting in and doing its job.
But of course, it's not quite that simple. We can get even more specific. A full agonist is like flipping that light switch all the way up. You get the maximum possible effect. A partial agonist is more like a dimmer switch. It turns things on, but only part of the way. And then you have inverse agonists, which are kind of weird and really cool. See, some of our body's receptors are naturally a little bit on all the time, even with nothing there. An inverse agonist comes in and forces that switch into the off position,
actively shutting down that baseline activity.
So let's say we've found it. We have a molecule that's the right size, the right shape, and it does exactly what we want it to do.
That's great, right?
Well, that's just the very beginning. How does that promising little molecule survive the incredibly long, expensive, and difficult journey to actually become a medicine you can buy?
We're talking about a journey that takes, on average, more than 10 years and can cost billions, yes, billions with a B, of dollars. It all starts in the lab with drug discovery. If a molecule looks promising, it moves on to preclinical tests, usually in animals, just to see if it's safe. And only if it clears that hurdle can it even think about starting the long, multi-year process of clinical trials in humans, all under the watchful eye of the FDA.
And make no mistake, this process is an absolute gauntlet. The stats are just mind-blowing. Out of all the drugs that make it to the starting line of human trials, less than a third will ever cross the finish line and get approved. The failure rate is massive at every single step.
The human testing part is broken down into 4 phases, each answering a different crucial question. Phase 1 is all about safety. They use a small number of healthy volunteers just to make sure the drug isn't toxic. If it passes, we go to Phase 2. Now we're asking, does it actually work? This is tested on a small group of patients who have the disease. If the answer is yes, we move to the giant Phase 3. This can involve thousands of patients, and it's all about confirming on a huge scale that it's safe and effective. And even after all that, after it's approved, the work isn't done. Phase 4 is basically forever. It's ongoing surveillance to catch any long-term problems out in the real world.
So yeah, it's a long, expensive, and incredibly tough process. But it's not just red tape. It's a system that's been carefully built and refined over decades. And it's all there for one reason, so that when you open your medicine cabinet, you can have confidence that what's inside is both safe and effective.
So that's the story of how a drug gets made, today. But this whole field is changing, and it's changing fast. We have these powerful new tools now, like AI, that can design and test new molecules in a computer, and genomics that let us create drugs tailored for your specific DNA. It really makes you wonder, doesn't it? What is this decade-long gauntlet going to look like for the amazing medicines of tomorrow?